So, this is part 3 of chapter 43 on the immune system. And we left off with the function of a fever when a person is infected. So, the benefits of a fever are the result of an infection. So, what happens is activated macrophages actually release a cytokine that's called interleukin 1. And that interleukin 1 is a chemical messenger that travels to the hypothalamus, telling it to increase the body's thermostat and raise the body's temperature. Because a higher temperature helps the immune system in a number of ways. It can inhibit the growth of bacteria. It can stimulate the leukocytes to increase their phagocytosis. It can speed up the repair of tissues. As we know, it speeds up enzyme action. It can cause the liver and the spleen to store iron, which is an interesting roundabout way to actually attack bacteria, because by reducing the level of iron in the blood, bacteria aren't able to get the iron that they actually need to grow. One other thing I want to mention before moving on is sometimes you hear the phrase septic shock. And what that is when certain bacteria actually can induce a system-wide inflammation response. So that response we looked at in the previous slide here, where we have an inflammation and the release of histamines and the capillaries being dilated, this entire process of inflammation, when it gets activated and really overstimulated throughout the body, that is called systemic inflammation. And that's where we get this idea of a septic shock. This is a result of this system-wide kind of overwhelming bacteria infection. Some of the characteristics of that type of septic shock is a high fever. So again, we have the macrophages releasing lots of that interleukin 1. A low blood pressure because of that dilation of the capillaries. And it actually leads to death. It's a very serious result of a system-wide or body-wide inflammatory response. So now the third line of defense is our lymphocyte defense, our acquired or active immunity. So this is a very specific line of defense that is mounted by our B cells and T cells, which are also known as our B lymphocytes and T lymphocytes. And then the B lymphocytes are also responsible for providing the immune system with antibodies, which are also called immunoglobulins. So those are the main characters or players in the acquired immunity. They are developed, they actually are formed in two different places. We'll look at that in a second. But their function, they start out as what are called naive B cells or naive T cells, which means they haven't yet encountered an antigen. But once they do, they will respond to a specific antigen, and that will be the antigen that they always respond to in the future. And that an antigen is any specific pathogen that causes and presents itself in a way. It's a foreign molecule that is specifically recognized by the lymphocytes and elicits a response from those B cells and T cells. That's the definition of an antigen. And B and T cells bind to that antigen via a protein in their membrane, their cell membrane, that's called an antigen receptor. So antigens can be any number of things. They can be pathogens, so bacteria, viruses. They can also be toxins like poisons, or they can be an abnormal body cell, something that's undergone some sort of change, such as in cancer. So how are invaders recognized? Through these antigens. So these antigens are proteins that serve as a name tag. So they're found on the outside of a cell, and they're kind of the calling card or name tag of that cell. So foreign antigens cause a response from our immune system. And again, they can be viruses, bacteria, protozoa, parasitic worms, fungi, toxins, even pollen, or tissue that is not normal to self, which would be some sort of transplanted tissue. So B cells and T cells respond to different antigens. The difference between how this response occurs is that B cells only recognize an intact antigen. So they have to see the whole antigen. So either they're going to see that antigen floating around in the blood or the lymph system. Whereas T cells can recognize and respond to just a fragment of an antigen. So this means these antigens have already entered a cell, so a normal body cell, and infected that cell. And then that cell is then going to show fragments of the antigen, and the T cells will recognize that. So again, B and T cells are lymphocytes, and where they develop, they come from the same stem cell line, but they develop in different locations. So B cells, and where we get the B, is from the fact that they come from bone marrow. So that's where they mature and develop. And they, again, because they're looking for the entire antigen, they are considered the humoral response system. The word humoral comes from humors, which means body fluids. And so B cells are floating around in the body fluids and looking for antigens that are floating around in the body fluids. B cells, another characteristic of them is that they're responsible for producing antibodies. Now, T cells, the T comes from the fact that they mature in the thymus. So the T is for thymus. And they are considered the cell-mediated response system, which means they're focused on infected cells. So they're not looking for an antigen floating around the body's fluids. They're actually looking for infective cells. So they're the cell-mediated response. And again, I mentioned before that until a lymphocyte encounters an antigen, it's considered a naive lymphocyte. So a naive B cell or a naive T cell means they haven't learned about antigens yet. So B cells, these are again just reiterating some of their key characteristics. They're involved in the humoral response. They defend against attackers that are circulating freely. They have a very specific response. They produce specific antibodies against a specific antigen. And there are two types of B cells. There are the plasma cells. These are the ones that are producing the antibodies. They are circulating. They have a rapid response to an antigen, and they have a short-term release. The second type of B cell is called a memory B cell. So when this system, the lymphocyte system, gets activated from an invasion from a pathogen, there are the production of these B cells and the release of these B cells throughout the body, or the actually multiplication because they're already circulating. And a certain portion are going to become very specific for attacking that particular antigen that's part of a pathogen. Some of those B cells are also going to become memory cells, which means they're going to remember this particular antigen for the future. And so if it ever presents itself again, they'll be ready to go and ready to attack it. Now, again, B cells are involved in producing antibodies. So, what is an antibody? An antibody is made up of protein and it's designed to bind to a specific antigen. So, that means one particular antigen for one type of antibody. It's made of multiple chains of proteins produced by the B cells, and they actually have this Y shape which the Y becomes the binding area. And that's the variable area of an antibody because it varies according to the type of antigen it's going to bind to. So we can have millions of foreign invaders and foreign antigens, which means we have to have millions of antibodies. Every antibody is unique and specific, and what makes it unique and specific is its binding area. So just like an enzyme has a very specific binding area, and we have this idea of a lock and key where only a specific key is going to fit in that lock, it's very similar with an antibody where we have a very specific site where only a very specific antigen is going to fit. This is a close-up of what it might actually look like. If we think of these different amino acids making up a protein, this is our antigens Y-shaped. Not the antigens, our antibodies Y-shaped binding site. And then this is the actual antigen here that's going to fit into that site. And this, another definition you should be familiar with is the portion of the antigen that actually fits into the binding site is called an epitope. The E-P-I-T-O-P-E, that may be a little hard to read right there, is an epitope. And that is simply the portion, the small portion of the antigen that will bind into the receptor site on the antibody. Also, there are receptor sites on the B cells, and there are receptor sites on T cells. All of these are going to bind the epitope of an antigen. Every B cell has about 100,000 antigen receptors, but each of those antigen receptors on a single B cell only bind one specific antigen. So they're not a bunch of different types of antigen receptors, they only are going to bind one type. Here's a close-up structure of the antibody, and you can see this Y shape. So the variable region, is this very top portion here, and this is called the light chain portion of an antibody. And then the bottom portion is called the heavy chain. And these antibodies actually are both embedded in the cell membrane of a B cell, and they also can be found floating freely. So you can see here we have these antibodies that are bound into the membrane of these B cells, and they can also be found floating free in the bloodstream. So they can actually function in two different ways. And this is again a more graphic three-dimensional picture of what an antibody is going to look like. How they work, how antibodies work, is in one of four general ways. So an invading pathogen gets tagged with antibodies. And one of the things that can happen once it gets tagged is that then a macrophage will recognize those tags and engulf and destroy the pathogen. So this is called neutralization, of the invading pathogen.